Melanism is a development of the dark-colored pigment melanin in the skin or its appendages and is the opposite of albinism. Historically, it was also the medical term for black jaundice. The word melanism is derived from the Greek mu epsilon lambda alpha nu omicron sigma. Pseudo-melanism, also called abundism, is another variant of pigmentation, characterized by dark spots or enlarged stripes, which cover a large part of the body of the animal, making it appear melanistic. A deficiency in a total absence of melanin pigments is called a melanism. The morbid deposition of black matter, often of a malignant character causing pigmented tumors, is called melanosis. For a description of melanin-related disorders, see melanin, melanosis coli and ocular melanosis. Adaptation. Melanism related to the process of adaptation is called adaptive. Most commonly, dark individuals become fitter to survive and reproduce in their environment as they are better camouflaged. This makes some species less conspicuous to predators, while others, such as black panthers, use it as a foraging advantage during night hunting. Typically, adaptive melanism is heritable. A dominant gene, which is entirely or nearly entirely expressed in the phenotype, is responsible for the excessive amount of melanin. Adaptive melanism has been shown to occur in a variety of animals, including mammals such as squirrels, many felines and canids, and coral snakes. Adaptive melanism can lead to the creation of morphs, the most notable example being the peppered moth, whose evolutionary history in the United Kingdom is offered as a classic instructional tool for teaching the principles of natural selection. Industrial melanism. Industrial melanism is an effective urban pollution prominent in many species of arthropods. It is the phenomenon of an organism evolving dark pigmentation when exposed to an environment polluted by dark soot deposit and sulfuric buildup from industrial pollution. In this type of industrial melanism, the darker pigmented individuals develop a higher fitness and are favored by natural selection. This change in favoritism as a result of modification in selection pressure is one of the best noted cases of Darwinian evolution. The most common case of this adaptation is found in the arthropod order Lepidoptera. This order of insects encompasses all insects with microscopic scales on the wings, such as moths and butterflies. Some species thought to undergo the selection of industrial melanism are Adelia bipunctata, Hamadryas feronia, Odontoptera bidentata, Oligia latruncula, Oligia stridulus, and many other species of moths, but probably the best and most studied example of industrial melanism is in the peppered moth Biston betulillaria. Originally, peppered moths lived where light-colored lichens covered the trees where they would rest. To camouflage with the tree, they originated with a light-colored pigment that helped them to avoid predators by not being a visual distraction. Lichens are great bioindicators of air pollution. Sulfur dioxide present in the atmosphere reduces the amount of lichen covering the surrounding trees. Eventually, industrial pollution came to the area, and sulfur dioxide began to kill the light-colored lichens off the trees, exposing the dark bark of the moths' resting places, creating a drastic contrast in color and making the light-colored moths more vulnerable to predation. This slowly altered the balance of the population, as light-colored moths were not surviving to reproduce and pass on their genes to the next generation, and the darker-colored moths began to breed more due to the newfound rise in fitness. This reversal of survivability made the gene contributing to melanic forms of the moth more desirable. The melanic phenotype of the Biston betulillaria has been calculated to give a fitness advantage as great as 30%. As generations went on, Eventually the population of peppered moths transformed from the majority being light-colored moths to the majority being dark-colored moths. Melanic betularia have also been widely observed in North America. In 1959, 90% of the betularia in Michigan and Pennsylvania had the melanic phenotype. By 2001, melanism dropped to 6% of the population, following clean air legislation. The drop in melanism is also correlated with an increase in species richness of lichens, a decrease in atmospheric sulfur dioxide, and an increase in the pale phenotype. The return of lichen and reduction of atmospheric sulfur dioxide is directly correlated and helps illustrate how influential air pollution levels are to the frequency of melanism for betularia. In each of these situations, the melanized form of the organism was found in an environment in correlation to higher amounts of industrial pollution found in the area. Many studies have been done to determine the reasons for the favoritism of darker pigmentation. Most studies aim to define the mechanism as a visual camouflage factor that gives the darker pigmented individuals a higher fitness, but some studies try to find other ways that melanization can be an asset and, in turn, more favorable than the lighter pigmented individuals. It is thoroughly noted that higher populations of darker pigmented organisms develop when there is an increase in industrial pollution in the area, but the relationship cannot be fully proven because the exact reason for increase in survivability cannot be tracked and pinpointed. To further solidify the theory of industrial melanism as an actual selection factor within evolution, surveys of melanic population rates and changes in atmospheric condition have been reported to establish and confirm trends of favoritism of darker, pigmented individuals in an industrially polluted area. As government regulations of air quality control have been implemented throughout the United States and the United Kingdom, researchers have collected data on melanic populations before and after the decontaminating effects of these regulations. According to the results, melanic population rates are consistent with the theory of industrial melanism, meaning that the level of melanic population was directly correlated to the abundance of contaminated air quality. When the levels of atmospheric pollution were high, so was the population rate of dark-colored moths, and when the levels of atmospheric pollution were low, there was a decline of melanism in the population. Industrial melanism is mostly concerned with the visual effects of melanization. The idea is that the darker pigmentation helps to camouflage the organisms better in an environment darkened by pollution and therefore heightens the fitness by making it less accessible to predators. But there are few other ideas about why melanization can help improve fitness in air-polluted areas for these populations that are not correlated to any visual effect at all. It is theorized that organisms that experience melanization have better immunity to toxic chemicals put into the environment by industrial pollution. Other studies have been conceived to find other aspects of melanization contributing to the survivability of the organism other than visual inconspicuousness. In the black archer's moth, it has been determined that the darker pigmented forms have also developed a stronger immune response to foreign objects with the attribution of the melanic pigment, the same melanic pigment that contributes to the visual coloration of the moth, is also involved in the encapsulation process of foreign invaders through the immune defense of the dark-colored form of the organism. This enhanced defense against foreign invaders is helpful in an environment contaminated by industrial pollution because the toxins that are put out into the air have less chance of negatively affecting the inhabitants of that environment. Another proposed mechanism for rapid industrial melanization is that there is a thermal advantage from the darker coloration. Many suspect that this thermal advantage, coupled with the predation advantage, gives species such as the bistron betularia and Adelia bipunctata increased fitness. The thermal advantage of industrial melanism is directly linked to the pollution aspect of industrialization. Smoke and particulates in the air reduce the level of sunshine that reaches the habitats. The bistron betularia and Adelia bipunctata melanic phenotypes become selectively advantageous in this situation because the dark coloration allows for a more efficient absorption of the limited sunlight. In colder environments, the thermal advantages of industrial melanism increases activity and the likelihood to mate. In the Netherlands, melanic Adelia bipunctata had a distinct mating advantage over the non-melanic Adelia bipunctata. In the Adelia bipunctata, copulation is much more likely to occur with warmer body temperatures and increased activity. Increased body temperature also gives melanic morphs a competitive advantage in searching for food because they are more active. Industrial melanism allows species such as the bistron betularia and the Adelia bipunctata to more efficiently warm the bodies in environments of reduced sunshine, increasing the mating activity and nutrition, thus increasing their fitness. In felines, melanistic coat coloration occurs as a common polymorphism in 11 of 37 felid species and reaches high population frequency in some cases but never achieves complete fixation. The black panther, a melanic form of leopard, is common in the equatorial rainforest of Malaya and the tropical rainforest on the slopes of some African mountains, such as Mount Kenya. The serval also has melanic forms in certain areas of East Africa. In the jaguarundi, coloration varies from dark brown and gray to light reddish. Melanic forms of jaguar are common in certain parts of South America. In 1938 and 1940, two melanistic bobcats were trapped alive in subtropical Florida. In 2003, the dominant mode of inheritance of melanism in jaguars was confirmed by performing phenotype transmission analysis in a 116 individual captive pedigree. Melanistic animals were found to carry at least one copy of a mutant MC1R sequence allele, bearing a 15 base pair in frame deletion. 10 unrelated melanistic jaguars were either homozygous or heterozygous for this allele. A 24 base pair deletion causes the incompletely dominant allele for melanism in the jaguarundi. Sequencing of the agouti signaling peptide in the agouti gene coding region revealed a 2 base pair deletion in black domestic cats. These variants were absent in melanistic individuals of Jeffroy's cat, Wanzilla, pampas cat and Asian golden cat, suggesting that melanism arose independently at least 4 times in the cat family. Melanism in leopards is inherited as a Mendelian monogenic recessive trait relative to the spotted form. Pairings of black animals have a significantly smaller litter size than other possible pairings. Between January 1996 and March 2009, leopards were photographed at 16 sites in the Malay Peninsula in a sampling effort of more than 1,000 trap nights. Of 445 photographs of melanistic leopards taken, 410 came from study sites south of the Kra Isthmus, where the non-melanistic morph was never photographed. These data suggest the near fixation of the dark allele in the region. The expected time to fixation of this recessive allele due to genetic drift alone ranged from about 1,100 years to about 100,000 years. Melanism in leopards has been hypothesized to be causally associated with a selective advantage for ambush. In birds, in April 2015, an extremely rare black flamingo was spotted on the Mediterranean island of Cyprus. Immune system. Melanin has several physiological roles in maintaining health, such as the synthesis of vitamin D. Melanin is the primary determinant of the degree of skin pigmentation and protects the body from harmful ultraviolet radiation. Synthesis of 1,25-dihydroxyvitamin D3 in the skin, however, is dependent on ultraviolet B light. Highly pigmented skin, to the level found in people of African origin, abrogates almost all ultraviolet-induced 1,25-2-D3 synthesis. Numerous animal models and clinical studies have underlined the essential role of vitamin D as a modulator of the different processes of the immune system. Evidence indicates that serum concentrations of 1,25-2-D3 and the prevalence of autoimmune diseases in a certain population are associated with the latitude at which the population resides. Genes for melanism in felines may provide resistance to viral infections. A viral epidemic may explain the prevalence of black leopards in Java and Malaysia and the relatively high incidence of black leopards and black servals in the Aberdare region of Africa. Previously, black-furred felines in the Aberdare had been considered a high-altitude adaptation, since black fur absorbs more heat. Studies reported in New Scientist magazine in 2003 also suggested that recessive gene melanism is linked to disease resistance rather than altitude. Melanistic cats may have better resistance to disease than cats with normal color codes. This would explain why recessive melanism persists when melanistic individuals are disadvantaged because they are poorly camouflaged in open areas. Sociopolitics. The term melanism has been used on Usenet, internet forums and blogs to mean an African-American social movement holding that dark-skinned humans are the original people from which those of other skin color originate. The term melanism has been used in this context as early as the mid-1990s and was promoted by some Afrocentrists, such as Francis Cresswell Singh. Bibliography. David Attenborough, The Life of Mammals. United Kingdom. BBC. Kettlewell, Bernard. The Evolution of Melanism. Clarendon Press. ISBN 0-1985737007. Majerus, Michael. Melanism, Evolution in Action. Oxford University Press. ISBN 0-1985498222. Melanism and Disease Resistance in Insects. Fry, 2013. How should the history of industrial melanism in moths be interpreted? The Linnaean, 29-1522.